Welcome everyone. My name is Mina Nabavi, and I'm a program manager with the Education Core with the UAB Counteract Research Center of Excellence in Arsenicals. Welcome to today's Grand Round seminar on the UC Davis Counteract Center of Excellence, Improving Medical Countermeasures for Acute Intoxication with Seizurogenic Chemical Threat Agents. For those of you not familiar with our center, it is led by Dr. Mohammed Athar, and the goal of the center is to develop mechanism-based, highly effective medical countermeasures against arsenical chemical warfare agents. I wanted to share a couple of upcoming activities and events within our center. The UAB Research Center of Excellence in Arsenicals is one of the sponsors of this year's Center for Clinical and Translational Science Bioethics Forum, which will be held virtually on Friday, February 18th from 11 a.m. to 1 p.m. The theme of this year's forum is Translational Science and the March to Health Equity. Registration information is available on our website, and please plan to attend this important forum. For those of you involved with the NIH Counteract program, there has been an update to the summer symposium dates. So due to the new Juneteenth federal holiday, the NIH Counteract Network Research Symposium has been moved one day to Tuesday, June 21st through Thursday, June 23rd. NIH will be sending a formal change of date announcement soon. Registration will open in March, and we will send the registration link when it is available. For more information on our center or its activities, please visit our website at uab.edu/arsenicals or send us an email at as33@uab.edu. On our website, you will find links to recordings of past Grand Round presentations and to our podcast. So our speaker today is Dr. Pamela Lein. Dr. Lein earned a BS degree in biology from Cornell University, an MS degree in environmental health sciences from East Tennessee State University, and a PhD in pharmacology and toxicology from the University of Buffalo. She completed postdoctoral training in molecular immunology at the Roswell Park Cancer Institute in Buffalo, New York. Currently, Dr. Lein is a professor of neurotoxicology and chair of the Department of Molecular Biosciences in the School of Veterinary Medicine at the University of California, Davis. Dr. Lein's research focuses on the cellular and molecular mechanisms by which environmental stressors, including persistent organic pollutants, organophosphorus cholinesterase inhibitors, and traffic-related air pollution contribute to the pathogenesis of neurodevelopmental and neurodegenerative disorders. Her research has been continuously funded by the NIH for over the past 20 years, and she has over 225 reviewed publications and book chapters. Dr. Lein is actively engaged in teaching and mentoring veterinary graduate and undergraduate students in neuropharmacology and neurotoxicology. Her service activities include director of the Career Development Program in the NIEHS-funded Environmental Health Sciences Center at UC Davis, director and PI of the NIH-funded UC Davis Counteract Center of Excellence, and co-editor-in-chief of the journal Neurotoxicology. So welcome, Dr. Lein. We're so glad you could join us today.
I really appreciate that introduction, and I really appreciate the opportunity afforded to me by the UAB Counteract Center of Excellence to present the work that we're doing here at UC Davis. I'm looking forward to a really interesting exchange at the end of my presentation. So I've been speaking for less than a minute yet, but yeah, you've probably realized I speak rather quickly, and when I get excited, I can go even faster. So please do not hesitate to ask a question through the chat. I've already given Mina permission to interrupt me and let me know if somebody has a question of clarification along the way. And I think if we can just leave it to questions of clarification during the talk so we have time for questions and answers at the end, that would be awesome. So you already seen the title for my talk today. Again, I'm Pam Lein, and just to acknowledge that this work was funded by the Counteract program through the National Institutes of Health. In particular, our program is funded by the NINDS. And the project also used core facilities that were supported by the UC Davis Mind Institute Intellectual and Developmental Disabilities Research Center, which is funded by the NICHD. There we go. Okay, so in the UC Davis Counteract Center, we're really focused on chemical threat agents that trigger seizures. And in particular, we're focused on two mechanistically distinct groups of seizurogenic chemicals, the anticholinesterases, which are typified by the organophosphates, and the GABA receptor inhibitors. And a sort of exemplary example of that are the TETs rodenticides. These compounds at high enough concentrations and acute exposure can trigger seizures in humans. That's been documented. And in humans, those individuals who survive the acute seizurogenic effect, which can lead to status epilepticus, a life-threatening condition, often go on to develop neuropathology, depression, anxiety, cognitive deficits, or acquired epilepsy. This has also been shown now in extensive animal literature. So the long-term center goals at UC Davis are to identify improved treatments for seizures and lethality caused by acute intoxication with these seizurogenic chemical threat agents, and also to develop therapeutic strategies for mitigating the seizure-induced brain damage that we see in those individuals who do survive. For today's talk, in the interest of time, I'm not going to present all our work on the tetramines, TETs, but I will be focusing on the organophosphates. So this is just a quick overview of the organization of the UC Davis Counteract Center of Excellence. I'm not going to spend a lot of time on this, other than to say that we have 3 research projects. The first project is developing and leveraging unique in vitro model systems to identify novel mechanisms of damage and to screen for potential therapeutic activity. The second project is really focused on identifying more effective anti-seizure agents to really prevent and terminate the acute effects of acute intoxication with these chemical threat agents, whereas the third project is focused more on those long-term effects. We are supported by 3 scientific cores. The first core is our metabolomics, pharmacokinetics, and immunoassays core. As you can see here, they perform serum assays for pharmacokinetics. They do metabolic profiling to identify protective mechanisms, and they're actively developed and actively involved, I should say, in developing nanobody-based methods for detection of threat agents, in particular tetramine. Our second core is the synthesis, screening, and medicinal chemistry core. So they're involved in synthesizing convulsants. So it's not possible to buy TETs, so we have to synthesize it in-house. They also synthesize a number of the novel convulsants that we've been screening in our center. And then also a number of other anti-inflammatories that I'll briefly touch on at the very end of this talk. And then, of course, we have a third core that is basically our statistics core. So the accomplishments of UC Davis Counter Center over the past 10 years, we're currently in our 10th year of funding, includes over 150 peer-reviewed publications. As I said, we've been very involved in models development. So during the first 5 years of the center, we spent a lot of time developing models, characterizing models, and then also developing quantitative approaches for really looking at the outcomes in those models. We've developed in vitro and in vivo experimental models for assessing the pathogenic mechanisms of, as I said, both the organophosphate cholinesterase inhibitors as well as the GABA receptor antagonist, and then using those models to start to screen for responses to therapeutic candidates. We've also spent a lot of time and published a number of papers on quantitative imaging approaches for assessing neuropathology and therapeutic efficacy in these models. These include MRI, PET, and micro-CT modalities that can be readily translated to the clinic, as well as semi-automated higher throughput methods for quantifying neuropathology and brain sections. We also, our biostatistics core has developed a number of advanced statistical models for analyzing complex data sets, including with animal measures. And then, as I indicated earlier, Bruce Hammack in one of the cores has been successful in developing a number of antibodies against tetramethylene disulfyltetramine, or TETs, and has really leveraged those nanobodies to develop ELISAs for quantifying TETs in biological and environmental matrices. And then we've also recently identified meclamylamine, which is a non-specific nicotinic antagonist, as a candidate prophylactic for acute OP intoxication that has a better therapeutic index than pyridostigmine. Unfortunately, in the interest of time, I won't be able to go into detail on any of these, but these are described in those peer-reviewed publications that have come out of the center. Additional accomplishments include the identification of soluble epoxide hydrolase inhibitors as candidate therapeutics for mitigating the long-term neurological sequelae associated with acute OP intoxication. I again won't really be spending much time on this today, but this is a focus of work moving forward. And then today I will be talking about the identification and advancement of Alopregnenolone as an in-field anti-seizure treatment for rapidly terminating both OP and TETS-induced status epilepticus when administered up to 40 minutes post-exposure. And for today, I'll really be focusing more on the OPs than the TETs. So the OP that we use in the center is diisopropylfluorophosphate, which you'll see abbreviated as DFP throughout my talk. The structure of DFP is shown here in the upper left. This is considered a credible terrorist threat agent by the United States Department of Homeland Security, which is one of the reasons we've adopted it for our studies. It's a very potent neurotoxicant, and similar to the OP nerve agents, which we don't have authorization to use here at UC Davis, it has a very similar structure and principal mechanism of action. In other words, inhibition, very acute and rapid inhibition of cholinesterase in the brain. As you can see here, when it does inhibit cholinesterase, this results in excessive cholinergic signaling within the central and peripheral nervous systems, and acute intoxication with DFP rapidly leads to seizures and respiratory distress. And if therapeutics are not provided or therapeutic interventions, it will cause death. This is the rat model of acute DFP intoxication that we spent a lot of time in the first 5 years really kind of refining and characterizing. And so you can see here that DFP is administered to the animals at 4 mg per kg subcutaneously. And then within a minute, we have found that we have to administer atropine and 2-PAM in order to keep the animals alive. Within several minutes after exposure to DFP, the animals begin to show signs of acute cholinergic crisis, otherwise known by the acronym SLUD, which stands for salivation, lacrimation, urination, and defecation. Within another minute or two, they start to develop tremors. Within 5 to 8 minutes, they begin to show classic signs of status epilepticus, including forelimb clonus, rearing of the torso, forelimb and hind limb clonus, rearing and falling, and then true status epilepticus usually starts within 8 to 10 minutes of exposure of DFP. In our classic studies that we use to really screen for therapeutics or assess therapeutic efficacy, typically we are administering therapeutic treatments about 40 minutes after exposure to DFP. The reason for this is that, as many of you know that are affiliated with the Counteract program, the Counteract Center, NIH, is really interested in therapeutics that can be administered in a civilian mass casualty. So in other words, therapeutics that can be administered at delayed times after the exposure, because all the scenarios that have been worked out basically have come to the conclusion that it will be 20 minutes or more before emergency personnel will arrive on the site of an exposure if it's really in a large urban setting. So as you can see down here in the bottom trace that shortly within 8 to 10 minutes after the DFP administration, we start to see status epilepticus as shown by EEG, so electrographic seizures in addition to behavioral seizures. And this is maintained for up to 4 hours post-exposure. So this is just again illustrating for you in a little more detail that acute DFP intoxication in this animal model will trigger electrographic and behavioral seizures. The electrographic seizures are shown on top here, so the timeline is along the bottom. So we start recording EEGs about 30 minutes before administration of DFP, and then continue to record for the next 4 hours. And you can see within about 10 minutes, as I indicated earlier, we start to see an increase in spike activity and spike frequency and amplitude. And each of these little dotted boxes, A, B, and C, are blown up in the trace above, so you can see them in more detail. And again, you see a lot of spikes with very high amplitude and very high frequency. This dotted box here, A prime, is blown up to the left, so you can really see the spike wave activity, which is very indicative of status epilepticus. And then as time goes on, we can see that the nature of the spikes changes. So here at about almost 2 hours out, we see an increased amplitude of these spikes. So the frequency is maybe a little bit different. And then this is still continuing up to 4 hours later. In terms of the behavioral assessments of seizure activity, we use a modified Racine scale to score behavior. So a score of 0 refers to no signs or symptoms. 1, the animal is exhibiting SLUD, or those symptoms of acute cholinergic crisis. A score of 2 indicates muscle fasciculations and tremors. A score of 3, forelimb clonus. 4, rearing hind limb clonus, and 5, rearing and falling with loss of riding reflex. A score of 3 or above is considered indicative of status epilepticus, and indeed in our correlative studies where we compared the behavior to the electrographic activity, we see there is a very tight correlation. In panel C here on the right, you can see the mean seizure scores for, in the case of the vehicle, which are the open circle, 17 animals. In the case of the DFP intoxicating animals, which are the black squares, 21 animals. You can see the mean seizure score at any particular time after exposure to DFP is 0 for the vehicles, as we would expect. And then the DFP animals, very quickly we see these scores climbing to 3 or above where they're maintained, certainly about 2.5 up to 4 hours later. One of the things that we discovered in the center is the classic paradigm that was used by most of the cataract world and investigators outside of cataract looking at OP-induced seizures was to pre-administer pyridose stigmine bromide about 10 to 30 minutes before administering DFP. This is the classic prophylactic that's used by the Department of Defense for military personnel entering into a theater of war where they expect that potentially there might be chemical threats employed. And so a lot of researchers have adopted that. It's also was thought at the time to provide better protection of the animals so they would survive the acute status epilepticus, and then you could look at long-term effects. We did a study to determine if pyridose stigmine treatment was actually required because, of course, in the case of a civilian mass casualty, civilians are not likely to be pre-treated with pyridose stigmine. So the question is whether that was necessary for this model. And so what you can see here in terms of the average seizure score, that we had an average seizure score over the 4 hours of about 2.5 in the animals that had no pyridose stigmine bromide treatment, and that was not varied in those animals that were pre-treated with pyridose stigmine bromide. Importantly, if we looked at the percent survival at 24 hours post-exposure, again, we see that having pyridose stigmine bromide on board did not make a difference. It's also been shown in our lab, and I don't have the data here, but if the animals survive during those first 24 hours, almost 90% of them will go on to survive for months after the exposure. So the bottom line was that pyridose stigmine pre-treatment did not seem to enhance survival in the rat DFP model or change the characteristic of the seizure activity. Therefore, moving forward, we no longer use pyridose stigmine bromide pre-treatment in our model. So one of the things I said at the beginning is that the human literature has very clearly documented that humans that are acutely intoxicated with OPs at levels that cause acute cholinergic crisis and or lead to status epilepticus show persistent neuropathology. So one of the critical things for us was to demonstrate this was also true in our model. And so what you're looking at here are representative photomicrographs of brain sections from vehicle animals versus animals treated with DFP 28 days post-exposure. In the columns here on the far left, we're looking at nu-N staining. Nu-N is a biomarker for neurons. And what you can very clearly see if you look at the brown staining here is that we have a very nicely defined hippocampus. And if we look in the animals that were treated with DFP, we see that the density of that staining seems less throughout the cell layers in the hippocampus, and that the upper blade of the dentate gyrus is totally missing. So there's been significant neuronal cell loss. In terms of neuroinflammatory biomarkers, which are another key characteristic that we see following acute OP intoxication, astrogliosis was measured by GFAP, immunohistochemistry, and microglial activation was measured by IBA1, immunohistochemistry. These are markers, of course, for astrocytes and microglia, respectively. Vehicle along the top, we can see that there are quite a few astrocytes, which is what you expect. If you look at the blow up here in the inset, you can see the morphology. These have nice sort of typical looking cell bodies and nice thin processes extending from them. In contrast, in the DFP exposed animals, we can see there's a very marked difference in the distribution of GFAP positive cells. And if we look at their morphology, they're swollen and engorged, and their processes have become very hypertrophic, indicating astrocyte activation. With respect to microglia, again, in the vehicle, we do see some scattered microglia throughout the brain section. Again, this is what you would expect. A higher magnification shows what is considered a resting state for microglia. So they have highly ramified processes in a relatively small cell body. If we compare that to the DFP exposed animal, you can see that the number of IBA1 positive cells has increased dramatically and that their morphology has also changed. They look really swollen and angry. We've done a series of spatial temporal expression profiles using these biomarkers, and I'm just showing you here the neuroinflammatory response. And if we look at microgliosis or astrogliosis as a function of time post-exposure, we can see that the areas that are affected as well as the intensity of the staining changes with time. So early, 12 to 24 hours post-exposure, IBA1 immunostaining is upregulated, and that's what this red color is. And you can see down here, minimal means a sort of a light color, whereas darker colors mean severe. And so we can see that there is microgliosis occurring across multiple brain regions. At 14 to 28 days later, we can see there's an increase in microgliosis, and that different regions are sort of differentially impacted, although all the regions that were initially impacted are just showing more severe staining, although the degree of staining varies between regions. By 60 days post-exposure, you can see that microgliosis is beginning to resolve in some regions, but in other regions it is not. Again, there's sort of this time and region dependent response in the profile microgliosis. We see a very similar sort of thing with astrogliosis, that it's region and time dependent. We've also developed PET imaging to be able to track neuroinflammation longitudinally in the same animal over time, which not only saves animals, of course, but is a more rigorous scientific design, because we can actually use an animal as its own control, because of course baseline neuroinflammation is going to vary from animal to animal. And so in this case, we're using ligands for TSPO. TSPO is a transmembrane protein that is not expressed at very high levels under normal physiologic conditions in the brain, but is strongly upregulated in microglia and astrocytes in response to neuroinflammatory conditions. And so what you're looking at here is the uptake of a radio-labeled ligand for TSPO. And the scale is down here in the bottom. So a score of 0.0 means no uptake, whereas a score of 2.0 means a significant uptake or significant upregulation of TSPO. And then this is color graded. So black means no uptake, whereas as these hot colors with yellow representing very high uptake. And so the brain itself, you can see the skull delimiting right here. So we see this bright area right outside the skull, which is the uptake of the ligand in sort of structures outside the brain parenchyma. And what you want to look at is the structures inside the skull, which is the brain parenchyma. And you can see there's very little labeling for TSPO in the vehicle. This has increased very significantly in the DFP intoxicated animals. We see a significant upregulation at 3 days. And again, this is somewhat region dependent. We have hot spots here in the hippocampus and down here in the piriform cortex. And we can see that we still have increased labeling for TSPO as much as 160 days out. So this is a very persistent neuroinflammatory response. Again, consistent with what's been observed in animal models of OP nerve agents and also in humans. We've also scored the behavior in these animals. We've looked at reactivity, general locomotor behavior, emotional behavior, and learning and memory at both 30 and 60 days. And what's you can note here is in terms of reactivity, these animals are hyperreactive as early as 7 days post-exposure, and this persists out to 60 days exposure. We see no change in the open field tasks, indicating no change in general locomotor activity. And then in terms of emotional behavior using the elevated plus maze, these animals that are treated with DFP seem to show anxiolytic behavior. They're less anxious. This has been noted in some populations of humans that have been acutely intoxicated with OPs. The big one, though, of course, is learning and memory. This has been documented in humans. We also see using Pavlovian fear conditioning that DFP-exposed animals have significant deficits in spatial and cued memory. The other thing that's been documented in humans that have been acutely intoxicated with OPs is persistent electrographic abnormalities. And in the DFP rats, we were the first to show that they also exhibit spontaneous recurrent seizures. You'll see abbreviated SRS in my slides. So about 50 to 70% of DFP intoxicated rats will develop SRS as determined using behavioral criteria, which I'm not showing you, but confirmed by EEG in a subset of animals. Here we're looking at a cohort of 6 animals, and 5 out of 6 animals developed these SRS. So here's a representative EGG in a DFP rat at 100 days post-exposure. So this is sort of the trace over, I think this is over several hours. And you can see that most of the time we don't see anything that would be indicative of a seizure. And that's shown here in the blow up in A. So this is sort of, if you will, background or baseline EEG. But every now and then these animals will trigger into a spontaneous recurrent seizure. And electrographically, this is indeed a seizure. This raster plot just sort of indicates in these 6 animals that were being monitored, how many SRS they were having over a 6-hour time frame. And this is the distribution of this. Some of these animals were having a lot of seizures over that 6-hour time frame, whereas others were having very few. So there's a lot of variability in the response. And again, it's somewhat dependent upon the time post-exposure. So one of the critical things that we've done in the Counteract Center at UC Davis is to really look at current medical countermeasures to determine how effective they are. And this was actually a charge that was put to us by the program officer of the Counteract program. And so the experimental paradigm that we did was to administer animals with DFP, followed again very closely with atropine and 2-PAM, and then 40 minutes after DFP to administer a benzodiazepine. In this case, we're using midazolam, which is the benzodiazepine of choice moving forward for the Department of Defense. The dose that we're using, 1.8 mg per kg, administered IM, is the rat dose equivalent to 2 of the 10 mg advanced anticonvulsant system autoinjectors that are recommended for use in humans. As you can clearly see here, that administration of midazolam does attenuate or reduce the amplitude of the seizures, but as shown in this blow up of these trace following midazolam exposure, it really does not terminate seizure activity. The current medical countermeasures also really don't prevent DFP-induced neurodegeneration. They do mitigate it, but they don't prevent it. And so you're seeing represented slides here of fluoro-JC staining, which is a marker for neurodegenerating, I should say degenerating neurons. And this is images of the CA3 subregion of the hippocampus at 3 and 6 months post-exposure. So there's very little to no fluoro-JC staining in the vehicle animals. This is strongly present in animals that were treated with DFP. In animals treated with DFP plus either diazepam, the benzodiazepine of choice prior to the acceptance of midazolam, or midazolam, we see significant fluoro-JC staining, which is attenuated somewhat by diazepam and somewhat by midazolam, but not reduced back to vehicle levels. So we quantify this using a semi-automated high-content imaging system, which removes a lot of sampling bias as well as interpretive bias in the data. And then our biostats core has developed this method of showing the quantitative analysis, which I'm going to walk you through because it's not typical. So what you see here is the geometric mean ratio of 2 groups being compared to each other. The circle is the actual geometric mean ratio, whereas the bars are the 95% confidence interval. If the ratio or if the outcome between the 2 groups being compared was similar, then the ratio should be 1. So the way to interpret these graphs is any comparison in which the 95% confidence interval does not include 1 indicates a significant difference, a statistically significant difference. And so if we're looking at these data, again, we have 3-month and 6-month time points here. Comparing the DFP animal versus the vehicle, we can see that DFP significantly increases fluoro-JC staining at 3 months. And I should also mention, I forgot to, that we looked at 7 brain regions: the amygdala, CA1, CA3, and dentate gyrus of the hippocampus, the piriform and somatosensory cortex, and the thalamus. And there were no significant region differences in these analyses, so all the data were collapsed across all 7 brain regions. But there were differences with respect to time, which is why the time is separated out. Now compare this and I guess I should point out, I guess we also see significant differences between DFP and vehicle at 6 months. If we now look at the what happens in the group that was treated with diazepam versus the group that got just DFP, we can see there's no significant difference. So in other words, treatment with diazepam does not effectively or significantly reduce the fluoro-JC staining following DFP exposure. The geometric mean ratio is lower, but the 95% confidence interval is above. So there's a trend towards reduction, but it's not statistically significant across the population. Similarly, midazolam does not in this case, midazolam does significantly reduce fluoro-JC staining. But if we compare diazepam to midazolam, there appears to be no difference. This maybe neuroprotective effect of midazolam is no longer apparent at the 6-month time point. So there appears to be a transient beneficial effect, but it's not maintained over that prolonged period of time. We also see that the current medical countermeasures have region-specific effects on DFP-induced astrogliosis. Here are representative images of astrogliosis in the thalamus at 6 months post-exposure as detected by GFAP immunoreactivity. The vehicle alone, so we see a baseline level of GFAP. We have the DFP animals, massive upregulation of GFAP immunoreactivity, still increased relative to the vehicle in the DFP animals treated with diazepam or midazolam, but maybe attenuated relative to DFP. And so here's that same sort of analysis that we saw before of the geometric mean ratio with a 95% confidence interval. In this case, there were no time-dependent differences, so the data are collapsed across the 2 time points, 3 and 6 months post-exposure. But there were region differences. So if we look first at DFP versus vehicle, we can see that in all brain regions, the level of GFAP is increased relative to the vehicle. If we look then at the other treatment groups, which are diazepam versus DFP, midazolam versus DFP, or diazepam versus midazolam, we really only see a few significant differences. One of these is that midazolam seems to reduce astrogliosis in the CA1 region and to reduce GFAP in the somatosensory cortex. So there are some significant neuroprotective effects, but they're very, very region specific. We see something a little bit different in terms of the effects of current medical countermeasures on DFP-induced microglial activation. Again, representative images, in this case in the thalamus at 6 months post-exposure. And we're looking at both IBA1 immunoreactivity, which is a biomarker for microglia, as well as CD68, which identifies phagocytic microglia, which are thought to be the very activated form of microglia. Again, DFP upregulates biomarkers for both of these outcomes. We see that DZP, diazepam, and midazolam seem to knock it back maybe a little bit, but not much, if anything. And there's still a lot of IBA1 staining. And indeed, if you look at the quantitative data over here, we see maybe a trend towards a decrease in microgliosis in animals treated with midazolam, but it does not reach statistical significance. This is an interesting one. So we also started looking at calcification in the brain using micro-CT. So micro-CT, basically we've shown in this model detects calcium mineral deposits. And these are present in the brain at 6 months post-exposure. So here's the 2D cross-section of the micro-CT image. And you can see some sort of lightening here, or hyperintensity. These are the areas of calcification being basically picked up by the micro-CT. We then do a 3D reconstruction of these areas of hyperintensity, and this is what the data looked like. And so we can see that at 6 months post-exposure, DFP is causing these, or leading to these calcium deposits. These maybe are attenuated by diazepam and midazolam, but they're still present. Again, quantitative data over here. So we have 3 month versus 6 month. We're looking at the dorsolateral thalamus on the top and the medial thalamus on the bottom. We can see that DFP compared to vehicle, we have an increase in calcification at 3 months. This is persistent at 6 months. Diazepam does appear to reduce calcification in the medial thalamus at that 3 month time point. That protective effect doesn't seem to persist out at 6 months. And we see really no neuroprotective effect of midazolam. So I hope I've convinced you that there is a clear need for improved medical countermeasures for treating acute OP intoxication, because the current medical countermeasures do not terminate OP-induced seizures when administered at delayed times after seizure initiation, and delayed administration of these medical countermeasures does not prevent long-term neurological sequelae. So we have focused in the second 5 years of funding on advancing Alopregnenolone as an adjunct therapy, in addition to midazolam, to rapidly terminate seizures when administered at delayed times after seizure initiation. During the Q&A, I'm happy to discuss the rationale for looking at Alopregnenolone as an adjunct therapy, but basically these are our marching orders from the Counteract program. So allopregnanolone, this basically is a neurosteroid. It is an endogenous neurosteroid that is produced in the brain. This is the structure of allopregnanolone, and it binds to GABA receptors in the transmembrane domain. And so by doing so, it potentiates the current, the chloride current that runs through the GABA receptor. In the interest of time, I'm not going to spend a lot of time on these particular graphs. I'm happy to come back to them later if anybody's interested, but they show that in our hands, in our in vitro models, we do see a potentiation of chloride current. Now, this is important, of course, because chloride is the major, or I should say GABA is the major inhibitory neurotransmitter in the brain. And so activating GABA currents, GABA chloride currents, will decrease activity in the brain to offset the seizure activity. So this is also how benzodiazepines work. So benzodiazepines bind to the GABA receptor and potentiate chloride channels. So why in the world do we think allopregnanolone would be more effective than just midazolam alone? Well, it turns out that benzodiazepines and neurosteroids like allopregnanolone bind to the synaptic receptors, which are, of course, opposite the presynaptic site of GABA release, whereas the neurosteroids also bind to the extrasynaptic receptors, which also are binding GABA. These extrasynaptic receptors are activated during status epilepticus because there's so much GABA being released that it's spilling over into this extrasynaptic site. One of the very well-established phenomena in status epilepticus, whether it's triggered by OPs, by TETs, whatever, is that there will eventually, and usually it's about a 20-minute period of time, be a desensitization of the synaptic GABA receptors. So these are internalized, which is why it's thought that these seizures become refractory to treatment with benzodiazepines. In contrast, the extrasynaptic receptors are still expressed, they're still functional, and we believe this is why allopregnanolone might be a really useful adjunct therapy for more effective seizure control. So when we did our initial tests with allopregnanolone, we actually looked at it in the presence or absence of Parampanil. Parampanil is an approved anti-seizure medication for children. It's an AMPA receptor antagonist, and it works basically by blocking glutamate flux into the postsynaptic cells. Glutamate, of course, is the major excitatory neurotransmitter in the brain. Excessive glutamate will trigger seizure activity. And in fact, this is the currently accepted paradigm for how organophosphates are triggering seizures in the brain. So by inhibiting acetylcholinesterase, they increase synaptic levels of acetylcholine, causing overstimulation of muscarinic cholinergic receptors, which is leading to an activation of glutamate receptors. And it's the prolonged activation of these glutamate receptors that not only are triggering seizures, but also resulting in status epilepticus. So we reasoned that by targeting both the inhibitory current to increase inhibition and targeting the glutamate or excitatory current to decrease excitation, that we'd have more effective seizure control than targeting either of those independently. And so the experimental paradigm that we used for these studies is shown here. We took a baseline EEG for 15 minutes prior to administering our DFP, followed of course by atropine and 2-PAM, and then our treatments were administered 40 minutes later. This is just our control situation to convince you that our vehicles that we used for these drugs were not having an effect on the seizures. So what you're seeing in this trace down here is the baseline EEG, which is just a normal EEG activity. When we put the DFP in, which is this red line, we can see that within a matter of minutes, we start to see very intense seizure activity, which persists for more than 10 minutes. So it's definitely status epilepticus. We administered the atropine and the 2-PAM. That really doesn't have any effect on the seizure activity. And then when we put the 3 vehicle injections in for the 3 drugs we're using, midazolam, atropine, and parampanil, we don't see any real effect on that seizure activity. So those are the control animals. And then if we look what happens when we start putting in midazolam versus midazolam plus aloe plus parampanil, we see some pretty interesting changes. So in panel A, we're looking here at the normalized EEG power over the first 40 minutes post-exposure. Here is our vehicle control across the top. So we see no change in that EEG power when the vehicles are injected 40 minutes later. When midazolam is put in 40 minutes later, we do see a drop in the amplitude of that EEG, as we showed earlier, but it never reaches back down to baseline. In contrast, when we put in a combination of midazolam plus aloe plus parampanil, and you can see the concentrations here, so 6 mg per kg of aloe and 2 mg per kg of parampanil, we see not only do the seizures start to terminate sooner, but they also go back down to baseline or below. If we look at this effect over a longer period of time, so here we're showing from 0 minutes to 4 hours, we can see a very similar situation persist over that 4-hour time period. And these are just the representative traces shown down here for you. Now, what happens if we don't use the combination of aloe and parampanil? So here we looked at just aloe plus midazolam versus just parampanil versus, plus midazolam. And you can see very interestingly that the aloe is much more effective by itself than the parampanil is. So we have a very rapid termination of the seizure, and it hovers just above the baseline for the first 40 minutes. And then it maintains that, although there's some recurrence of spike activity as you go out for 4 hours. In contrast, parampanil takes a longer period of time to drop that seizure power, EEG power, and it never really reaches baseline until much later. And you can see over here at about 100 minutes. In this case, it actually goes below the baseline, which is not necessarily a good thing either. So the other thing we did was look at behavioral seizures, and we saw the combined therapy is actually more effective than midazolam alone in terminating the behavioral seizures. So again, remember anything that's at 3 or above is considered status epilepticus. And we can see here that the vehicles alone really don't have much effect on that. These start to naturally wane after about 4 hours. And then if we look down here in the presence of animals that received the midazolam, we can see not much change in the behavior seizure score for the first 4 hours. If we look at the animals who received the combined therapy of aloe, parampanil, and midazolam, like we saw with the EEG, there's a very significant drop in the behavioral seizure score, which is maintained for 4 hours. If we compare aloe versus parampanil given alone with midazolam, we see what we saw with EEG. So the aloe in the blue bar, we see a rapid drop in that seizure behavior score, and that stays low for the first 4 hours. With parampanil, we do see a drop as well, also sort of back to the baseline level, but it takes a little bit longer. We did look at combined therapy versus midazolam alone in terms of mitigating those long-term neurological effects. I'm not going to go through the data for you. It is published if you're interested, except for the behavior where we have that's under review right now. But we looked at the following criteria. Do they decrease neurodegeneration, decrease astrogliosis, decrease microgliosis, decrease the frequency and or duration of SRS, and attenuate neurobehavioral deficits? And we're comparing combined therapy, aloe plus parampanil plus midazolam versus midazolam alone, and then monotherapy with aloe plus midazolam versus midazolam alone. With respect to the combined, we saw that there was no significant difference between the combined therapy and midazolam alone with respect to neurodegeneration. The combined therapy was superior to midazolam alone in terms of the neuroinflammatory biomarkers and in terms of decreasing the frequency and duration of SRS. However, it was not significantly different from midazolam alone in terms of attenuating those behavioral deficits. Interestingly, when we look at the monotherapy with aloe plus midazolam, we see that this is also superior to midazolam alone in terms of astrogliosis, and interestingly, also in terms of neurodegeneration. However, it was not in terms of microgliosis. We still don't know at this point which of these endpoints or neuropathological outcomes are driving the neurobehavioral deficits or the SRS. That's what we're currently working on now. But it is interesting to note that there are some differences between these different types of treatments. So despite the promise of combined therapy, we are pivoting to testing aloe alone as an adjunct therapy in the UC Davis Counteract Center. There's a couple of reasons. One is it's clear that there are going to be significant regulatory challenges in advancing a combination therapy versus a monotherapy as an adjunct to midazolam. Secondly, the scientific data suggests that aloe alone has a better therapeutic and safety profile than aloe plus parampanil. So our in vitro data, which I did not show you, demonstrates that parampanil suppresses all network activity, which is not a good thing. We don't need comatose people. And our in vivo data also indicated that parampanil decreases the EEG below baseline and is slower to terminate seizures than aloe. And again, this is the data I showed you earlier, just to remind you of that point. The other thing, as I said, is that the midazolam plus aloe appeared to be more neuroprotective than midazolam plus aloe plus parampanil. And there is some evidence to suggest that neurodegeneration contributes to those neurobehavioral deficits. And so here in the top graph, we're looking at fluoro-JC staining. This is a DFP animal treated with midazolam alone, DFP plus midazolam plus aloe versus DFP plus midazolam plus aloe plus parampanil. And you can see that there appears to be an attenuation of the yellow, which is the fluoro-JC staining, in the animals treated with midazolam plus aloe versus the other two conditions. And indeed, if we look at the quantification of the FJC per area, we can see that midazolam plus aloe versus midazolam is more effective in decreasing that parameter. And we can compare that to the combined therapy versus midazolam alone. We also looked at nu-N staining. So this is staining neurons. Here's what we see in animals treated with midazolam alone versus midazolam plus aloe versus midazolam plus aloe plus parampanil. And the critical thing here is if you look at the purple staining, which is a nu-N, you can see that that blade of the dentate gyrus is missing in the midazolam treated animals. It's present here in the animals treated with midazolam plus aloe, also present in the animals treated with the parampanil in addition to aloe, but not quite as many neurons as we see here. And again, that's kind of borne out over here. So if we, in this case, an increase in the geometric mean ratio is a good thing. It means there's more neurons. So here we have midazolam plus aloe versus midazolam showing a significant increase in that parameter. So the other thing that is important to know is that we've started to play with the dose of alopregnenolone because we thought, well, maybe 6 mg per kg IM is not high enough to really see the full extent of the benefits that might be afforded by alopregnenolone. So we've gone up to as high as 24 mg per kg, which seems to be relatively safe in these animals and actually correlates pretty well with some of the human doses that are being tried in clinical trials to treat refractory status epilepticus. And we can see here that in this case, if we look at the behavioral seizure score, the vehicles in black, midazolam treatment in the beige brown color here. If we look over here in the blue, these are animals that receive midazolam plus aloe at 24. And there's this really significant drop in that behavior seizure score that happens very, very quickly. Similarly, if we look at the cortical EEG, we can see that treatment with midazolam plus aloe at 24 mg per kg during that first 40 minutes rapidly drops the amplitude of the spike activity, and that is maintained over the first 24 hours. I'm sorry, first 4 hours. It also appears based on some of our preliminary in vivo imaging that aloe plus midazolam, so aloe at 24 mg per kg plus midazolam is more effective than midazolam alone in attenuating DFP-induced neuropathology. On the left here, you're looking at hemispheric T2 weighted images at approximately minus 0.5 millimeters of bregma. Anything that has sort of this hyperintensity relative to the vehicle indicates a lesion. And we can see that there are very prominent lesions in the DFP treated animals without therapeutic intervention at day 3, day 7, sorry, and day 28. These are the ventricles that are enlarged. We do see some hyperintensity here, for example, in the piriform cortex and in hippocampus. In the animals treated with midazolam, we still see hyperintensities that maybe are attenuated relative to the DFP animal without intervention. And that seems to be significantly attenuated in the animals treated with DFP plus aloe. And then we've also looked at the TSPO staining, again, at, or PET imaging, I should say, at day 3, 7, and 28. Very bright, hot colors here, the DFP animals. Those seem to be sort of attenuated in the animals treated with midazolam, and attenuated even further in the animals that are receiving aloe. And it appears that actually the combined therapy is probably the most effective than either one of these independently. I'm going to wrap up here pretty quickly. So I'm not going to spend a lot of time on these graphs. I think you will have the PowerPoint slides afterwards. But one of the things that's very interesting to note is when we start looking at the TSPO PET data and quantifying that we see that these interventions are differentially attenuating neuroinflammation. So we're looking at the standardized uptake value for the TSPO PET ligand and the amygdala, the hippocampus, the piriform cortex, and the thalamus at 3, 7, and 28 days. If we compare midazolam versus DFP, we really, see some protection here in some brain regions. So we see some in the hippocampus at 3 and 28 days. And again, here in the thalamus at 3 and 28 days. Not clear what's happening at 7 days, why it's not protective there. But if we look at that in contrast to Alopregnenolone versus DFP or midazolam plus aloe versus DFP, we see more protection in more brain areas. And this seems to be the most protective when we have the combination of midazolam plus aloe. What's really interesting here is showing that, again, as sort of indicated in this graph, is intervention with combined aloe, that should be aloe plus MDZ, is more effective than either alone. So here we're looking at aloe versus midazolam, the combined versus midazolam, and the combined versus aloe. And again, you see more of these significant blue bars here in the combined treatment. What's really important to note, though, is that even with all of these interventions, there is still persistent neuroinflammation in the DFP animal compared to vehicle. So if you look at the DFP animal versus vehicle, you can see all of these confidence intervals are statistically increased relative to the vehicle. If we look in the DFP animals treated with midazolam, we don't see any differences here. If we look at the DFP animals treated with aloe versus vehicle, we see some changes here in some brain regions at 28 days. And even in the combined, which has got the best safety profile or best therapeutic profile, we still see that it's not true across all regions, that it's really bringing it back down to vehicle level. So they're improving the situation, but they're not really decreasing it to background. So why is that? We're effectively terminating seizures. And the mantra in the field has been forever that if you can terminate seizures, and the quicker you terminate seizures, that you actually will protect brain. And so time after treatment equals the amount of brain that's protected. And so we think it's because we're treating late, so there is some damage that started, but also because we found that DFP can cause persistent neuropathology in adult male rats independent of seizure activity. And so we have found that about 10 to 13, 15% of animals treated DFP do not respond with seizures. And that's shown here with the behavioral seizure score. So they are showing seizure scores that are consistent with cholinergic crisis, but not consistent with seizure activity. And when we look at these, sorry, go ahead. When we look at these animals in terms of electrographic activity, I'm sorry, I clicked too fast there. We see that in the animals that are the low responders, there's very little evidence of electrographic seizure activity, whereas in the high responders, we see very, very pronounced electrographic seizure. Brain cholinesterase levels are the same in both the DFP and the high responders, so it's not because the DFP isn't getting to the brain. But we do see neurodegeneration. So at 1, 4, and 60 days post-exposure, nothing in the vehicle in terms of FLORA-JC staining. We don't see anything at 1 day in the DFP, low responder, but we do in the high responder. But by 4 days, we see there's very significant change. In the interest of time, I'm not going to go through the quantitative data. And again, the other really cool thing that we found is that regardless of whether they respond with seizure activity, we see some very significant mineralization in the thalamus at 60 days post-exposure, which doesn't seem to differ significantly between the low and the high DFP responders. So in summary, We basically have shown that the rat model of acute DFP intoxication recapitulates the acute and chronic effects of the acute OP intoxication observed in humans, that current medical countermeasures neither terminate seizures effectively nor adequately protect against chronic neurological sequelae. Adjunct therapy with allopregnanolone effectively terminates seizures, and it seems to attenuate subsequent neuropathology. However, that combined therapy with alloimidazolam does not completely protect against neuropathology. And observations of DFP low responders suggest that acute OP intoxication can cause neuropathology independent of seizure activity. So moving forward, we're assessing the safety of high-dose allo for use as an adjunct therapy for rapidly terminating seizures in the field, but also screening anti-inflammatory agents for efficacy as neuroprotectants. And I'd just like to end with acknowledging my collaborators in the Counteract Center, some of whom are shown here. So we have Mike Dorota, and the key people in his lab who contributed the data I showed to you today were Dorota Solkowska and Ashish Deer, my colleague Isaac Pesa, who's done all the in vitro studies, Bruce Hammock, who synthesizes TETs and some of our anti-inflammatories, myself, and the people in my lab who have been very instrumental in generating these data are listed here. Heike Wolf, who is an anti-inflammatory expert, and then our statisticians, Daniel Tangretti and Daniel Harvey, who are not shown in this picture. And with that, I'd be happy to take any questions that you might have.
Thank you, Dr. Lein. We had several questions come in right at the end. The first one was, do baseline levels of inflammation correlate with the severity and frequency of long-term neuropathology?
That is an awesome question. And we're trying to get our hands on that. We thought we had an answer to that, which was, yes, they do seem to be highly correlated. Certainly neurodegeneration and neuroinflammation are highly correlated both spatially and temporally, and in terms of severity. But those have not really been predictive of whether the animal will have SRS or will have neurobehavioral deficits. So we're still trying to parse that out. So it's a great question. And I think understanding that will be key to knowing which we have to target in terms of therapeutics.
Another question that came in, have you studied allo as a pre-treatment?
We have, although not to a very large extent, because that's not the type of research the Counteract program wants to fund. They're interested in post-exposure therapies. But if we give allopregnanolone pre-exposure, it can be very neuroprotective and actually prevent seizures from occurring. But allopregnanolone has a relatively short half-life, so it has to be administered relatively soon before the administration of DFP in order to be highly effective.
Another one, is there a gender difference in severity of OP-induced seizures and long-term deficits?
That is such an awesome question, and one that's totally understudied in the field. So we have studied that in juvenile animals. I did not present that today. We also have developed a juvenile model of acute OP intoxication, and we clearly see gender differences there. So the males have much more severe seizures than the females, and consequently, they also have much more severe neuropathology than the females. Interestingly, the neurobehavioral deficits are similar in both. So that's sort of making us question. We're currently doing studies right now in the adult animals, and we do see differences in electrographic seizures between males and females. Again, females seem to have less severe electrographic seizures, and we also are seeing gender differences or sex differences in therapeutic response to midazolam. Females seem to be responding better than males.
Wonderful. We're at the top of the hour, and I know others have other meetings or engagements to get to. Dr. Lein, there were several other questions in the chat box, so I hope I can send those to you, and maybe we can include some answers when we send out this webinar once it's
absolutely done. I'd be happy to
Perfect.
do that.
Perfect. Well, thank you so much for presenting, and thank you to all of our attendees. We appreciate you joining our Grand Rounds seminar today. Have a great rest of the day.